So welcome all to the new module of Biotechnology. An introduction to Biotechnology. What is biotechnology? It's a fusion of biology and technology. The term was used or coined by Karl Erking, a Hungarian engineer, in 1970. The biotechnology deals with exploitation of biological agents or their components for generating useful products and services for mankind. The term biotechnology came into popular use in mid-1970s as a result of the increased potential for the application of engineering techniques for molecular biology. The word seems to have been employed by the Leeds city in 1920, but in fact, biotechnological processes are nearly 5,000 years old, which began with the discovery of fermentation, consequent production of the alcoholic beverages like wine, beer, etc. Beginning with fermentation, use of biological process then experienced over changes over centuries. But greatest revolution till now commenced with 1970s and 1980s, when a product of interaction between science of biology and technology came to a wider existence. This relationship got the name biotechnology. Fermentation, antibiotic production, baking, brewing are included in old biotechnology, whereas the new biotechnology involves cell culture, cell fusion, bioprocessing, genetic engineering. Thus, biotechnology is the application of biological organisms, systems, or processes to manufacture and for service industries, comprising a number of technologies based upon increasing understanding of biology at cellular and molecular level. The techniques include recombinant DNA manipulations, monoclonal antibody preparations, tissue culture, protoplasm fusion, protein engineering, etc. The term biotechnology gains several definitions as from different groups, such as the British biotechnologist defines biotechnology as the application of biological organisms, systems and processes constituting biotechnology, while European Federation of Biotechnology defines biotechnology as an integrated use of biochemistry, microbiology, engineering science in order to achieve technological, that is, industrial applications of the capabilities of microorganisms, cultured tissue cells and parts thereof. Again, it has another definition by the US National Science Foundation as the continuous controlled use of biological agents such as microorganisms or cellular components for beneficial use. It is also defined by the Japanese biotechnology as technology using biological phenomena for copying and manufacturing various kinds of useful substances. All the definitions of biotechnology differ in their approach and content and emphasis, but the two features are common to all. One is utilization of the biological entities, their components or constitutions like enzymes, in such a way that the second point is that some product or service is generated which will only enhance the human welfare. So it is the utilization of biological products to constituents to make a product that will be helpful to mankind. The examples are given here, like the use of microorganisms for the production of chemicals at a commercial stage was prompted by war effort, that is during the First World War. Germans were forced to develop a technology for glycerol. Glycerol was needed for the manufacture of explosives. When their supply of vegetable oil was disrupted due to British naval blockade, the Germans tried microbial production of glycerol by yeast. They succeeded in increasing the yield to over 1,000 tons per month. Similarly, British resorted to acetone-butanol fermentation using Clostridium acetobutylicum due to German interference with the normal supply of these chemicals. Acetone was required for ammunition, butanol for artificial rubber. The First World War also left the citrus orchards of the countries like Italy in ruins. The citrus grew were not taken care of and hence were spoiled. This resulted in a great increase in the rate of citric acid which was extracted from citrus juice. As a result, the technology for citric acid production using Aspergillus niger was developed in 1923. Production of antibiotic penicillin by Penicillium notatum was discovered in 1930 by Fleming, but its commercial production began only during the Second World War. Now, the different branches of biotechnology, the medical biotechnology or red biotechnology, this branch deals with red biotechnology deals with the protection of human health by producing recombinant vaccines, monoclonal antibody, valuable drugs, technology for gene therapy, DNA fingerprinting, etc. White biotechnology or industrial biotechnology deals with large-scale production of variety of biochemicals from alcohol to antibiotics, production of enzymes like proteases, lipases, amylases, etc. Enzyme technology. Techniques for remodeling of existing protein or enzyme in order to enhance their efficiency is called protein or enzyme engineering. Mineral extraction through leaching from low-grade ore is called microbial mining, etc. is involved in industrial biotechnology. Environmental biotechnology involves bacteria being used for detoxification of industrial effluents in compacting oil spills, for treatment of sewage, and for biogas production. Animal biotechnology. It's an in vitro fertilization and embryo transfer in human and livestock. Production of transgenic animals for increased that is milk, then growth rate, disease resistance, etc. and for the production of valuable proteins in milk, urine or blood. It's called as molecular farming. Then there is plant biotechnology or green biotechnology, which deals with the production of the transgenic plants for producing plants resistant to the attack of pest, insect, then virus, herbicides, etc. Germplasm conservation through storage in liquid nitrogen, rapid clonal multiplication through meristem culture of many fruit and forest trees. Then blue biotechnology is a term that is used to describe marine and aquatic application of the biotechnology, but its use is relatively rare. What is the scope and importance of biotechnology? We know that it is an applied science and it has made great advances in the field of molecular biology and microbiology. Since the two areas have been developing so much in the coming years, it has been exploited in a variety of ways for both the production of industrially important biochemical, including enzymes, and for basic studies in molecular biology. The scientists are now diverting themselves towards biotechnological companies like Biogen, Citus, GeneTech, Hybridtech, etc., many famous scientists, including Nobel laureates like Walter Gilbert, opted to work for biotechnology companies. In world, USA, Japan and many other countries of Europe are leaders in biotechnological research as encouraged by industrialists. Biotechnology now plays a very important role in employment, production and productivity, trade, economics and economy, human health and quality of human life throughout the world. Many commentators are confident that by 21st century it will be the century of biotechnology, just like the 20th century's era of electronics. Importance of biotechnology to human welfare would become obvious from the following examples, like protection of human health, production of monoclonal antibody which is used for disease diagnosis, example that is venereal diseases, sexual diseases, hepatitis B and other viral diseases, cancer, etc. DNA and RNA probes, that is again for disease diagnosis like Kala Azar, sleeping sickness, malaria, etc. that is parasitic diseases. Then artificial vaccinations for inoculations, then rare and valuable drugs such as human interferon, insulin, etc. The technology for gene therapy for treatment of genetic diseases, identification of parents or criminals using DNA or auto-antibody fingerprinting are very accurate and reliable from even blood, semen, stains and hair roots, etc. are notable achievements in the field of biotechnology. Microorganisms are being employed since several decades for the large-scale production of variety of biochemical ranging from alcohol to antibiotics. In processing of food and feed, which is a major part of industrial biotechnology, the techniques for remodeling of existing. So the importance of biotechnology, you have learned it is very important in the protection of human health, then for production of biochemicals, then using the biological agents like virus, fungus, bacteria for the control of plant diseases and insect pest control. Bacteria is also used for detoxification, for combating oil spills, for treatment of sewage, for biogas production. Microbes have also been used for extracting metals like microbial mining. Then in vitro fertilization and embryo transfer techniques have permitted childless couples suffering from one or other kind of sterility to have babies of their own. Hormone-induced super ovulation, embryo splitting coupled with embryo transfer can be used for rapid multiplication of farm animals including cattle. Genetic engineering is being employed to develop transgenic animals which are resistant to certain diseases, capable of faster growth rates and more efficient feed conversion, and with the capacity to produce certain valuable biochemicals and to excrete them in their milk, urine or blood, from which they can be isolated and purified. This is known as molecular farming. In agriculture, rapid and economic clonal multiplication of fruit and forest trees, production of virus-free stocks of clonal crops, creation of novel genetic variations through somaclonal variations and transfer of novel and highly valuable genes for disease and insect resistance through genetic engineering have opened out exciting possibilities in crop production, protection and improvement. Biotechnology seems to have an unlimited commercial potential in view of its capability to generate an unlimited range of valuable and useful products, services concerned with virtually every aspect of human existence.